without further ado, it's wonderful to welcome you to this last CCSAW research seminar of the semester. I'm Georgia Mason. I'm director of the Campbell Centre for the Study of Animal Welfare. Those of us here in Canada acknowledge that we're on traditional territories and treaty lands of Indigenous people, Inuit people, and Métis people. And here in Guelph, of course, we're based on the traditional territories of the Mississaugas of the Credit. If you're not Canadian and you're online and you're thinking, why do Canadians always do this? It's part of a truth and reconciliation movement to really acknowledge the kind of dark and murky past of Canada's colonial history. And so it's just a little tiny recognition that the land we're on was not originally European, of course. If anyone wants to take this kind of a little bit further, have a look at some of the websites about Indigenous involvement in World War I, II, and the Korean War, because obviously November is a month of remembrance. And there's some interesting things out there. Yep.
There's a comment on the chat about the sound of being
Oh, yeah.
You might just start concentrate.
Oh, thank you. Okay. The owl is now under my nose. Is that better sound, people online? Is that better sound? Someone give me a sound. Great, thank you so much. All right, good, good. Okay, then it's also my great pleasure to introduce my friend and good colleague, Professor Hanno Würbel. Hanno is a professor at the University of Bern, and he directs the Division of Animal Welfare there. He's got a long and distinguished academic history, really bringing his understanding of animal welfare and behavior to bear on the use of animals in research. And he has a sort of critical lens where he uses what we know of behavior and welfare to ask, first of all, why do we always keep them in bear cages when we know that makes them weird? Second question, why are the cages all identical when the humans they're modeling is so heterogeneous? Third question, why on earth is biomedical research failing so badly to produce reproducible or translatable results? And then his current question is, hey, maybe we shouldn't be in a glass house throwing stones because animal welfare science itself isn't always perfect. So what can we learn about critiques of other disciplines that we can apply to our own work? Amongst his many publications and accolades include winning the ISA Creativity Award, winning a Center for, it's the John Hopkins Center for Alternatives to Animal Use 3 R's Award. And he's also got some fantastic publications, many of which you can see on Google Scholar, but he's been involved in some really important policy pieces like the ARRIVE guidelines and also the US's Guide for the Care and Use of Laboratory Animals. Okay, without further ado, I'm going to pass you over to Hanno, who's going to talk about the principles of good science as they apply to animal welfare science.
Well, thank you very much, Georgia, for this nice introduction. Thank you, first of all, for having me for this presentation. I'm currently sitting in a rather miserable hotel room. I mean, not miserable, it's just hot and tiny because I'm at a conference in Germany. And so I'm trying to do my best to give a lively presentation here because, of course, it's always a bit difficult to talk to your own laptop when you would rather talk to people. And I can only see you in the very tiny right corner of my laptop because otherwise you just cover up all my slides and I don't know what to talk about. So it's quite a bold title that I've chosen for this presentation here, Principles of Good Animal Welfare Science, welfare in brackets, because of course, much of what I'm talking about is about science in general or animal science in general, because there just isn't that much meta research going on animal welfare science, so that I could use all examples from animal welfare science. But I think that many or much of what I'm going to talk about is applicable to animal welfare science. And I'm going to give you some examples that underscore this to some extent. So when that's the first freeze. Right, okay. So what do we mean by good animal welfare science, or what do we mean by the quality of animal welfare science? So if you look up what quality means, then that's how good or bad something is. And so we can ask, like, what is good science? What is good animal welfare science? And there are obviously different answers to this question. Science that answers relevant question, is that what we're looking for? Or is it science that uses rigorous methodology? Or is it science that is morally responsible? And I would argue that good animal welfare science cannot be just one of those. You may ask relevant questions, but if you don't use rigorous methodology, your findings will be useless. If you're not using rigorous methodology, you may be wasting animals for irresponsible research. And so I would argue that basically good animal welfare science is relevant, rigorous, and responsible. It has to be all of those. But who is deciding whether animal welfare science is good or bad? Now, generally, whether the questions that we're asking are relevant, that's in the hands of the funding bodies or the experts that advise funding bodies on whom to give money. And we're generally assuming that if someone is ready to pay for a study, then the question must to some extent be relevant, at least for those who are funding that research. Whether the methodology is rigorous, that is usually in the hands of our peers. So peer review decides on whether or not we can publish our findings, and the fact that we publish, or the fact that we're allowed to publish and that our papers get accepted, means that other scientists thought that what we did was right and was good science, so that it's worth being published. And whether or not what we're doing is morally responsible, that's often in the hands of ethical review boards who review our study protocols, and especially in animal research, of course, ethical review boards play an important role in deciding whether or not the benefits of the research are sufficient to outweigh the harms that may be imposed on these animals in the course of research. Now, I'm going to focus on the rigor of methodology today, for one, because there is a lot of research going on this, and there are some substantial problems out there. And it's also, I think, the most interesting one, because it's the scientific community that is responsible for the rigor of methodology. So first of all, it's the scientists who use the methodology and decide whether or not to use rigorous methodology. And it's their peers who either accept or don't accept their research findings, depending on whether they consider them rigorous or not. Now, that something may be wrong with science in general, that is obvious when you look into the literature of meta-research. And even though there is not that much out there on animal welfare science, if you look at science in general, then there have certainly been loud voices voicing concerns over the rigor of methodology in science. And John Ioannidis was not the first one to criticize poor methodology or lack of scientific rigor, but he was certainly one of the loudest and most prominent, and that is certainly also due to the fact that he dared to publish a paper that was entitled "Why Most Published Research Findings Are False." Now, that's of course not what we want to hear. Neither the ethical review bodies want to hear that, because if they approve of research on animals that turns out to be useless because the results are false, then it cannot be responsible research. Now, this wasn't just a catchy title. He actually did go into details, and this is all based on a large stream of meta-research that he and his group conducted, when they came to the conclusion that most research findings are false for most research designs and for most fields. And by this, I imply that it may also affect animal welfare science. Now, since then, we're talking about something that is often referred to as a reproducibility crisis. And this is data from a survey that the journal Nature conducted some years ago, when they asked their readers whether they think that we do have a reproducibility crisis in science. And 90% of those who participated in this survey, they did think that there were issues with reproducibility, and more than 50% thought that there was a significant crisis in terms of reproducibility. Now, this term reproducibility crisis has certainly been fueled by spectacular cases of replication failure, and these are certainly the most famous ones. It's two pharmaceutical companies, and they did in-house replications of studies that have been published in prominent scientific journals. So they picked the good ones, the famous ones, and they tried to replicate these studies in-house, because often these studies that publish or that report on spectacular findings, they may pave the way for advances in medical development. Now, what they found was that in one case, in the study conducted by Bayer scientists, of 67 studies that they attempted to replicate, they could only replicate 36% of the findings, while 64%, 2/3 of the findings, could not be successfully replicated. And in the study by Amgen, it was even worse, that only 11% of 53 study outcomes could be replicated in their own hands. Now, what are potential causes of such poor reproducibility? And there are, of course, many, and that's what I'm going to talk you through in the course of this presentation, is a couple or some of the most prominent or most likely causes of poor replicability, all of which probably contribute to these issues, some more, some less, and there is certainly variation across fields of research. And I'm trying to figure out which of these causes are most relevant for animal welfare science. So to start with, of course, there may be issues with reproducibility and certainly with translatability or generalizability if the animal models are not properly validated, or if they are essentially poor models for the constructs being studied. Now, this is a major issue, of course, in preclinical animal research, where we often use animal models that are very distant from human beings in evolutionary terms, but also in their general biology, including behavioral biology. And it has often argued that mice may not be the best model to model diseases in humans. But in addition to this, besides actually the species gap between humans and mice, there are other issues, and these have been discovered more recently, namely that it's not just that these animals are different from humans, but it's also that we breed and house them in ways that are not very conducive to them modeling relevant states in human beings. And one of the issues is that these laboratory animals, these laboratory mice, they are highly artificial creatures. In most cases, they are highly inbred. They are almost completely homozygous. That's one thing. And the other thing is that they have been, of course, derived from wild mice, but the way in which they have been bred and housed resulted in animals that are essentially germ-free, and they are very far removed in their microbiota and the pathogens that they carry from wild mice. And interestingly, more recently, researchers have discovered that this is a major issue, given that these laboratory mice, the immune system of these mice doesn't really develop beyond the state of a newborn, that they have started to actually rewild these animals by transferring embryos into wild mice in order to restore the natural microbiota and pathogens, thereby creating so-called wildling mice. And there are already some successful studies showing that the translational power of these models is much better than the highly hygienic standard laboratory mice. Besides the genetics and the general biology, there are, of course, also issues with the types of housing conditions that these animals are housed under. And this is a picture from my very early steps in applied ethology when I did my PhD thesis on stereotypies in laboratory mice, basically discovering that most laboratory mice, when kept under these Bern standard laboratory conditions, develop abnormal repetitive behaviors. And this is a picture that depicts two different types of stereotypies in two different strains of laboratory mice. So from the beginning on, because I was working on laboratory mice, of course, it was never only an issue of animal welfare, but it was always, because these animals are used for science, for research, finding that the health and well-being of these animals may be impaired, of course, also raised concerns about the scientific validity of these animals for producing valid research. And of course, more recently, Georgia Mason and her PhD student Jessica Kate and colleagues, they found that we're not much further than that. And even today, conventional laboratory housing increases morbidity and mortality in research rodents substantially. And this, of course, is a major welfare issue, but it's also a major concern in terms of the validity of these model organisms. Besides the animals themselves, whether or not they're adequate model organisms, of course, there's also a question about the measures that are used in research and whether these measures are valid outcome measures for the constructs that they are meant to measure. And one of those measures or test systems that have been have come under attack recently is the forced swim test that you can see here, where a mouse is placed in a water bath, and then the time is measured, how long the animal struggles to find an escape before it gives up. And this test has been used and is still being used a lot as a test for measures of depression in preclinical animal research. And even though researchers, neuroscientists have long demonstrated and argued that this test has essentially lacks construct validity for depression or as an outcome variable for depression, it still continues to be used. And of course, by this, it has become an easy target for animal rights groups who didn't even have to complain about whether or not this is stressful and painful for these animals. They could simply refer to other scientists demonstrating that this test lacks construct validity and therefore shouldn't be used with animals. Now, what about animal model validity and animal welfare science? Now, compared to preclinical animal research, of course, we are often much closer to our target populations, and many of us actually work on target populations. So whether I'm studying laboratory mice in the context of a research laboratory, or others studying laying hens or cows, cattle on a farm, generally animal welfare scientists study populations that are representative of their target populations. And often they also study these animals in settings that are much closer to the conditions under which these animals are kept on farms or in laboratories. So to test both the populations, the study populations, as well as the study settings are representative, or certainly more representative of our target populations than this is normally the case in preclinical animal research. But when it comes to the outcome variables or the test systems that we're using to measure animal welfare, then things get slightly worse. And of course, one reason for this is that animal welfare is an extremely complex construct. Animal welfare scientists are still struggling with finding common ground on what they mean by animal welfare, and while some strictly consider measures of health and biological function, others use behavior, normal behavior, and deviations from normal behavior to measure animal welfare. And yet others argue that the only thing that is of interest to animal in terms of animal welfare is how animals feel. And so we should use measures about what and how animals are feeling. Now, of course, even those who are using measures of health or behavior, implicitly they use these measures also as proxy measures of animals' feelings, trying to use indirect measures of well-being by measuring things that can be objectively measured, because of course we all know that subjective feelings can't really be measured objectively. So we always need proxy measures. And this is, I think, where a lot of the trouble with the measures that we're using comes from. But recently, Liz Paul, Mike Mendel, Christy Nicol and colleagues from Bristol, they have made an effort to figure out whether different measures of welfare actually converge. And so they studied chicken under different conditions and measured a range of measures using measures of preference, using measures of judgment bias, and also using more traditional candidate welfare indicators such as measures of stress, and then explored or assessed whether these measures converge. And as you can read on the left, an excerpt from their abstract, that overall the different approaches did not converge to identify a precise state of animal welfare, indicating that all those different measures that are used by animal welfare scientists to actually measure states of welfare, they seem to measure different things. And so they concluded that further work is needed to establish which alternative measures of affective state might be more appropriate indicators of animal welfare. Now, one problem that may contribute to this is that animal welfare scientists often use rather subtle interventions, probably because many of us shy away from torturing animals. We don't want to be nasty to animals. We want to be kind to animals. That's why we're animal welfare scientists. But this may actually cause problems. Now, even if I don't know whether this actually was a problem in this particular study, simply by showing you that the two conditions that were meant to induce poor welfare and good welfare, on the one hand, the good welfare state, which is conditions that the animals generally prefer, is for laying hens, a relatively large pen with a proper dust bath, with a proper nest for egg laying, with elevated perches and enough space for these animals, while the generally non-preferred condition was a smaller pen without a dust bath, without an appropriate nest for nest laying, and with only a small perch. However, if you compare these conditions to the conditions, even actually good conditions for laying hens in aviary systems, you can see that there are many more animals in these pens and many more animals per square meter, and the conditions are probably much worse than in both of these conditions. And so the question is, well, are we really able to model poor welfare states versus good welfare states if we're using conditions that are so far off to the good side of animal welfare compared to the conditions under which these animals are normally kept? So to conclude from this, I do think that we are in many ways better off compared to traditional preclinical laboratory animal research, in that our study population and study settings are usually much more representative of our target populations and target conditions. But still, we have issues with our outcome variables that may not always be properly validated. And so it's difficult to assess how good research is if we do not know how good the measures that we're using to measure welfare are. And a problem that the interventions that we're using to validate these measures are often so subtle that it may simply be unlikely to find major differences if we do not use more discrete treatments. A second major issue that is certainly also an issue in animal research in general, but also in animal welfare science, is that we're often using sample sizes that are too small and so lack statistical power. Here are data from a recent systematic review and meta-analysis across many studies in the neurosciences and metabolism research. And they found that most animal experiments are severely underpowered. So in this case, they analyzed almost 500 studies with more than 2,500 effect sizes. And as you can see, the median power achieved across all of these studies is just 18%. So normally we're aiming for a statistical power of about 80%. 18% means then less than 1 in 5 studies was sufficiently powered to detect an effect, even if an effect was there. And that's certainly not a good situation. Now, one of the reasons for this is probably that in most cases, people don't really do proper power analysis or proper sample size calculations. And this you can see here on the right side of the picture, where the numbers of experiments are displayed and how many of those experiments used how many animals in their studies. And as you can see, the largest number of experiments used 10 animals per group. So the median group size is 10 animals per treatment group. And I think this is probably not due to the fact that all these sample size calculations arrived at a sample size of 10 animals per group to achieve proper power, but this is just by convenience and by tradition. Researchers tend to use groups of 10 animals, maybe sometimes 12, maybe sometimes 8 per group, but this just does not reflect proper sample size calculations, and therefore many studies may end up using too few animals than they should. So how is that in animal welfare science? I mean, of course, as with all animal science, there is a certain pressure to use as few animals as possible. That's part of the 3 R's principles. But still, we're not meant to use too few animals, because if we're using too few animals, then our findings may be meaningless. And these animals, how few they ever are, may have been spent or wasted on inconclusive research. Now, this is a recent systematic review and meta-analysis on all the different studies that have been conducted using judgment bias as a measure of welfare in animals. And as you can see here, there were many studies across many different animal species. And this is all these results that you can see here are meta-analysis across studies using these different species. Now, you can see that all of the summary effect sizes of the species-specific meta-analysis, they all include 0, meaning that none of them found a significant summary effect size. If you combine them all together and do calculate the summary effect size across all of those studies, then you will find an effect size of 0.2 in terms of Hedges G, which is a small effect size, and it's barely significant. Now, these authors concluded that in individual empirical studies comparing 2 means to achieve a power of 0.8 at an alpha of 0.05, it would be necessary to have sample sizes of at least 50 animals per group for detecting moderate effect sizes. And by moderate, they mean an effect size of 0.4, which is twice as large as the one that they found across all of these studies. And this is certainly not the number of animals that are usually included in animal welfare science projects. So either we need measures that are more sensitive to the treatments that we're using, or we would have to use many more animals in order to achieve sufficient power. Another major issue that is general across research is the analytical flexibility that we have. As you know, in contrast to clinical research and clinical trials, we do not have to pre-register our study protocols, and so we can adjust our analysis and the decisions which results to publish in the end. We can postpone this until we have seen our results and can then decide on which results we find most interesting. And of course, this may lead to biases in the literature. And one of the problems is p-hacking. P-hacking meaning that when you have multiple outcome measures and when you can divide your study sample into multiple subgroups, you often have many possibilities for doing tests, statistical tests. And the problem is that if you do not correct for multiple testing, then with an increasing number of statistical tests, the chances to find at least one significant result by chance increases dramatically. As you can see here in the graph on the left side, with only about 10 to 15 statistical tests, you are already above 50% chances to find at least one significant result simply by chance. Another problem is harking, when the hypothesis, harking is short for hypothesizing after the results are known, when you adjust your hypothesis to the results after you've seen the results. And again, this leads to a bias in favor of false positive findings. And we have been looking for evidence on harking, and we haven't really found any empirical evidence on harking. But then in the course of a systematic review that Marianna Rossa, a PhD student in our lab, conducted on behavioral tests of anxiety in mice, we found some outcome measures that had really extremely wide distributions of the outcome variable to both sides. As you can see here, an example of the effect of diazepam, an anxiolytic drug that does not only have anxiolytic properties, but also sedative properties on locomotion in an open field test. So when we saw results like these, we wondered, well, how can it be that the outcome of a study is so widely spread, and yet the authors tended to interpret their findings either in terms of an anxiolytic effect or a sedative effect of the drug. And so we looked into this a bit more closely, and what we can see here is data based on a systematic review, including 151 studies, testing for the effect of diazepam on locomotion in an open field test. Now, all the effects that are colored in blue were reported as the drug had an anxiolytic effect. So these are the reported effects found in the results section, while those who found a decrease in locomotion, here the negative numbers, those who found a decrease in locomotion, they interpreted the effect of diazepam in terms of a sedative effect. Interestingly, there were some that found that even an increase in locomotion was evidence for a sedative effect, while some even interpreted a negative effect on locomotion as an anxiolytic effect. But what is much more interesting and puzzling, and in our view can only be explained by harking, is the fact that these effects have been predicted by these scientists. So when we looked at what the researchers predicted in the introduction or in the method sections of their papers, those who found an anxiolytic effect, so an increase in locomotion in the open field, they also predicted the drug to have an anxiolytic effect in most cases, while those who found a depressive or a sedative effect, they already predicted that in the introduction or method section. And interestingly, those who didn't predict anything, they didn't find an effect on either side of the scale. And I don't know if you have a better explanation, but we really did not find one other than that these predictions have been made post hoc after the researchers had seen these results. And then, of course, there are many other risks of bias. We all know the commands of good research practice, what we learn as students in undergraduate courses when we design studies, that we should randomize our study animals to the treatment groups, that those who treat the animals and those who assess the outcomes should be blind to treatment, that outcome variable inclusion and exclusion criteria should be defined a priori rather than after you've seen the results. So that's all standard knowledge to scientists, but there is some evidence that many scientists don't seem to adhere to these commands of good research practice. It's only indirect evidence, again, based on meta-research, and this is an example of a systematic review on preclinical animal studies of stroke. And what they did, they basically just analyzed papers for whether or not these measures to prevent risks of bias were reported in the method sections or not. And as you can see, those who reported, for example, that they used blinding and that outcome assessors were blind to treatment, they found much smaller treatment effects on average than those who did not report blinding in the method section. So even though we don't know whether they simply didn't report but actually did blind, this is highly unlikely given that those who did not report blinding did find such larger effects of their drugs compared to those who did report that they used blinding. So it seems that scientific rigor reduces the effect sizes discovered in science. And of course, this has implications for the evidence landscape. So whenever we do systematic reviews, meta-analysis of the evidence, then of course it is important to know under what conditions these studies were conducted. And it's very likely that due to the fact that often these measures to prevent bias are ignored and not adhered to, that the effects that we find in the literature are substantially overestimating the true treatment effects. This is also the reason or what instigated the NC3Rs in the UK to start developing reporting guidelines for animal research, because in so many papers you didn't find anything written about whether or not these measures against risks of bias were used. And so that's where the ARRIVE guidelines for animal research come from, that educated or reminded scientists what they should report in their papers. And of course, the aim was not just that scientists would report these, but they would actually take these into account when designing and conducting their experiments. But I think Adrian Smith was right, the head of Norecopa in Norway, that in principle, what we need are design guidelines rather than reporting guidelines if we want to make sure that study protocols are up to speed. And so they developed the so-called PREPARE guidelines, which they now promote by stating that you should prepare before you arrive. Now, how about risks of bias in animal welfare science? This really is an area where we don't have much empirical evidence, but there is some. And this paper from 2014, a group of Belgian researchers, they studied observer bias in animal behavior research, in particular, whether blinding has an effect on the results that we find, or expectations, if we know the treatments that animals have been exposed to, if that affects what we're observing. And they did some interesting studies, and here is one result where they showed the same videos twice to a group of people. So they trained observers to score positive and negative social behavior. And then they they gave them videos which they gave them in two versions, where they slightly changed the light and the illumination, but it was essentially the same videos scored twice by the same observers. But one time they told them that one group were control animals, while the other group were animals that were bred for high social breeding value, meaning that they are particularly friendly animals amongst each other. And as you can see here, that those videos that were labeled as animals with high social breeding value, they got higher scores on the side of positive social behavior and lower scores on the side of negative social behavior compared to the control videos, even though it was exactly the same videos that were scored by these people. And I think this is a good example demonstrating that unknowingly and unconsciously we're just affected by knowing what treatment animals have been exposed to, and the expectations that we associate with this can taint our perception or our tendency to score one or the other if in doubt. Now, often, in particular, animal behavior scientists and also animal welfare scientists, they often complain that blinding is difficult. You use several persons to collaborate. It's often complicated. Sometimes it's even impossible because you can see what treatments the animals are exposed to. But at least in those cases where it is possible to be blinded, I think we should all take the effort to do it. And here is a very recent study that has only recently been published as an uncorrected proof in PLOS Biology by Natasha Karp and colleagues, also in collaboration with the NC3Rs in the UK, where they did a qualitative study of the barriers to using blinding in vivo experiments and suggest improvements. And all those who are skeptical towards blinding and whether that's feasible in the context of their research, I recommend this publication highly. And the last point that I want to go into some detail is the generalizability that we achieve with our study population. And this, of course, is a particular issue in laboratory animal science, where the animals are usually bred and housed to be highly standardized. So they're usually genetically identical, and they are identical in terms of the conditions under which they are bred and housed. And this rigorous standardization may actually compromise the generalizability of the results. And because independent replicate studies are always different to some extent in the conditions under which they are conducted, this lack of generalizability may also compromise the replicability of study findings. Now, the interesting thing here is that while all those other causes of poor reproducibility that I've been talking about so far are actually considered as, or are known to be bad for scientific validity and replicability, while on the other hand, rigorous standardization is recommended in many textbooks as the thing to do when you want to achieve highly precise and replicable study findings. So in order to convince you that standardization is actually a cause of rather than a cure for poor reproducibility, I'll go back in history sometime to the birth of reproducibility as a key principle to establish scientific evidence. And that was back in the 17th century, when the Royal Society in England decided that from now on, in order for an observation or a scientific finding to be accepted as scientific evidence, it had to be replicated independently by an independent scientist. Now, of course, we've come a long way since then. And nowadays, we're not asking researchers to conduct independent replicate studies before they're allowed to publish their findings. But what we do instead is that instead of just observing one animal or one cage of animals, of course, we're using a sample of animals, assuming that this sample, the individual animals within this sample, that they constitute our independent replicates and therefore produce findings that are reasonably robust and replicable. But of course, if you look at this picture, it is evident that these animals are a lot less independent of each other than animals would be in independent replicate studies. So these animals, they usually share the same genotype, they share the same environment, they are handled by the same personnel, and the experiment is conducted on them by the same people. So they have a lot of shared genotype and shared environment, and therefore are much more similar to each other than animals from independent replicate studies. And I think the first time that this became, that scientists became aware of the issues with this was a study conducted by a group of behavioral geneticists in 3 different laboratories, where they compared different strains of laboratory mice in a range of different behavioral tests. And here is just one behavioral outcome measure from an elevated plus maze test, a standard anxiety test. And as you can see, there are some really robust differences between some strains here, as for example, between this HA strain and the C57 Black 6 strain, where you can see that there is some variation across the 3 laboratories. But in all 3 laboratories, there is a substantial difference between these 2 strains. On the other hand, the ones that I've colored here, you can see that in one laboratory in Albany, the difference is significant in one direction. In Portland, it was significant in the opposite direction. And in Edmonton, there was no significant difference between these 2 strains. So 3 fundamentally different conclusions drawn from exactly the same experiment. And these researchers, they went to great lengths to make sure that the conditions across these 3 labs were as rigorously standardized as is possible. How about, anyway, I put the word despite here in inverted commas, and this was actually the point that I was making after I had seen this paper, namely that the reason why they did find such laboratory-specific effects was not despite the rigorous standardization, but because of it. Because, of course, there are things that you can standardize across laboratories, but there are also many things that you simply cannot standardize, like the personnel that is interacting with the animals, the different smells in the laboratory, the noisescape, and so forth. So there are always things that will differ between laboratories, and the more you standardize the conditions within laboratories, the more these differences between laboratories will affect the results that you get from such an experiment. So that gave birth to the standardization fallacy, which I first formulated in a little letter to the editor, pointing out that actually standardization is maybe a cause of poor replicability rather than a cure. And since then, I've conducted with my lab and with my colleagues a lot of studies on this. We've recently published a statistical account of the standardization fallacy. We've done simulation studies on real data from preclinical research showing that if you combine multiple studies into virtual multi-laboratory studies, that the results of these studies are much more similar and much better replicable compared to single laboratory studies, where you find huge variation across studies. And we've also conducted a workshop with experts from different fields of research and came up with a perspectives paper on suggestions how we might actually incorporate biological variation into our study design rather than excluding it by standardization without needing to use more animals for our studies. So how about standardization in animal welfare science? Is that really an issue? There's certainly good news that our study animals are usually more diverse than the typical laboratory mouse. Our study settings are usually less standardized, but still and everybody who has studied animals in different pens, in different barns on different farms, they know that there are pen effects, barn effects, farm and study effects, and they can sometimes be very large, and they certainly need to be taken into account. And I completely agree with Christian Navroth and Lorenz Gigax, who recently stated in a paper on replicability replicability issues in animal welfare science, that studies in farm animal welfare science can take advantage of the heterogeneous farm settings that can serve as the population for sampling and accordingly follow a framework that improves external validity and thereby also increases replicability. So it's a bit like with clinical trials. Clinical scientists who often were jealous of the laboratory animal scientists, because laboratory animal scientists, they are in control of everything and they can control all the conditions of their animals as well as their genotype. But actually, it's an advantage if you want to obtain robust and rigorous results. Then, of course, you have to address the biological variation that is there anyway. And otherwise, you will just produce case studies, typical N-of-1 studies, where a single genotype is studied under a single environmental condition, and you cannot expect that this will produce robust and replicable findings. So if you look at all these different causes of poor reproducibility that I've been talking about here, we can actually group them into the 3 different types of validity that are important for the scientific validity of our research, and that is construct validity, internal validity, and external validity. And from this, I was tempted to propose the so-called 3 V's principle, of course, in analogy to the 3 R's, because whenever we conduct a harm-benefit analysis on the response, whether an animal experiment is responsible, we of course have to apply the 3 R's principle in order to minimize the harm imposed on the animals before we actually assess whether the benefit of the research outweighs the harms. And I would argue that we should also more systematically assess to what extent these 3 V's, construct validity, internal validity, and external validity, are guaranteed to maximize the scientific validity of the research and avoid wasting animals for inconclusive research. I'm very happy that Switzerland has taken this up, and in our newest version of the application form for animal experiments, researchers now have to detail why they think their study protocols are suitable to answer the questions that they're asking by giving some thought to the construct validity, internal validity, and external validity, as well as reproducibility of their expected findings. But there is more to responsible animal research than simply scientific validity and implementation of the 3 R's. You all know that we have entered an era of open science, and I would strongly argue that only open animal research can be responsible animal research. Here are some data from comparing studies that have either been pre-registered, so where the study protocol has been pre-registered before the study has been conducted, in comparison with the traditional literature. And what is shown here is the proportion of null findings, so the proportion of results that were not statistically significant. And as you can see, in those studies that were pre-registered, the proportion of null findings is much, much higher than in the traditional literature, suggesting that there is a massive publication bias against negative, non-significant results in the non-pre-registered traditional literature. And of course, if the negative findings don't get published, then we have a massive publication bias, and every systematic review and meta-analysis will be massively biased towards positive findings rather than true findings. Besides pre-registration, some of you may have heard about fair data management plans. Make sure that your data are findable, accessible, interoperable, and reusable by other scientists. Use preprint service to get your results out as soon as possible. Follow reporting guidelines and study protocol guidelines, and make sure that you publish open access, because as I said, I do believe that only open research can ultimately be responsible animal research. So with this, I would like to thank you for your attention and acknowledge some of my lab members who have contributed massively to those data from my own lab that I've presented, and some of my long-standing collaborators with whom I have worked on these issues for many years by now, and of course those who have given me money and thought that at least my research questions were relevant to be studied. So I'm happy